So this will be a quick review for Chapter 3 in Exam 1. So remember, the structure of the plasma membrane is that it is a phospholipid bilayer, and that it remember that phospholipid bilayer made up of a polar hydrophilic head and a nonpolar hydrophobic tail, meaning this one loves water, this one hates water, and that when you put them in a watery environment, that the water-loving heads will point out and the hydrophobic tails will point in. So that gives the cell membrane its classic phospholipid bilayer with the cholesterol embedded. You have proteins embedded, and then you have receptors made out of carbohydrate or lipids, glycolipids or glycoproteins, glycocarbohydrates that are for cell recognition. And so that gets to the 5 function of the cell membrane. And you can see that down in figure 3.5, that they act as transport, enzymatic activity, signal transduction, cellular recognition, and intercellular joining. But remember, to get the full 6 points, make sure you give a little description of what that means. With question 2, remember, passive transport does not require energy and moves with its concentration gradient, whereas active transport requires energy and moves against a concentration gradient. So you need to have both of these points for each type of transport to get full credit for that question. Question 3, where it's going to ask about 3 types of passive transport. So we have our simple diffusion, which allows for nonpolar and gases to pass across the membrane directly. We have facilitated, where you need the carrier or the channel proteins. And so you require a protein to move substances across. And those are going to be larger or charged particles. And then lastly, figure 3.8 is osmosis, where the water moves to equilibrate against the solute, not the solute, and that's usually through that semi-permeable membrane like we have with our plasma membrane. Questions 4 and 5 can be easily viewed in table 3.1. Same with the previous questions, you can find the answers to that. So our 3 types of passive: simple diffusion, facilitated osmosis, definition, examples. Active transport is our primary active transport. Common example is our sodium-potassium pump. And then when the concentration gradient is built and then it flows back across and carries a secondary molecule with it, this is called secondary active transport. So there are your list and describe 5 types of membrane transport. Table 3.1, our simple, facilitated osmosis, active transport, and secondary active, with a brief description for full credit. And then the 4 types of vesicular transport. Remember, we have 3 types of endocytosis: phago, pino, and receptor mediated. Here's the definition for each, examples of how each is used, and then the final exocytosis, releasing of the substances. So exiting is exocytosis, entering is endocytosis. And for question 6, why discuss, make sure you discuss the importance of active transport in the sodium-potassium pump. And you can see this here in section 3.3, and it talks about that 10 times higher than the concentration, these gradients are required for skeletal muscles to contract, the heart to beat, nerves to send impulses, cells to maintain their osmotic balance. So the steep concentration gradient created by the sodium-potassium pump is essential to body functioning. So this is important because it helps maintain a steep difference in concentration of these 2 substances, which is critical in the heart pumping, the muscles contracting, the nerves sending impulses. So very important to bodily function that we maintain that concentration gradient that is built by the sodium-potassium pump, which is a form of active transport. Module 3.4 will go over your cellular organelles, but at the end of that, you can find Table 3.2 that does a very nice job of summarizing the organelles. So describe the structure and function of the organelle. More importantly, the function. So you need to tell me about the organelle and then the function that it carries out within the cell. And that can be found in Table 3.2. And then right above 3.2, where it's describing the importance of the endomembrane system, you can find that at figure 3.21, and it provides a summary of the endomembrane system function. And there are 4 outcomes, 4A, 4B, 4C, that will result in the endomembrane system. Question 9, describe the structure and function of the 3 components of the cytoskeleton. You can find that in Module 3.5 and nicely summarized in Table 3.3. Again, make sure you give the functions. Talk a little bit about the structure, but mainly tell me about these functions. So describe the process of transcription and translation. Question 10. You can see this in figure 329 and 331. Both are initiation, elongation, termination. With transcription, begins with the promoter on the gene, then the RNA polymerase binds, opens up the DNA, adds the nucleotides, and starts creating the new messenger RNA transcript. Once it reaches the termination, transcription ends, and the messenger RNA is released. In your e-text, you can see the big picture animation here. Again, remember you can find all of these in the study area from the mastering area in Blackboard. Once the messenger RNA is made, it is modified before it leaves the nucleus, with introns being removed and the exons spliced back together. That is the mature RNA. And then that mature RNA comes out into the cytoplasm, joins with a ribosome. So this is translation, figure 3.31. You have initiation, where you have the ribosome, the tRNA, and the messenger RNA all coming together with the start codon. Then the ribosome moves 3 nucleotides or a codon, brings in the next. Via the tRNA, the amino acids join via a polypeptide bond, and then this continues until you reach, and this is elongation, continues until you reach termination when you get to a stop codon, which causes the whole assembly to fall apart. And again, big picture of translation, and you can find all these videos in the study area. So the details with 10, 11 explain the roles of the rRNA, messenger RNA, and tRNA. We just kind of went over those. These are the functions of each of those. And then with question 12, label the picture below. It's basically the same thing as our big picture animation, which is figure 3.32. So transcription, RNA polymerase catalyzes formation of our pre-messenger RNA from the DNA template. Then we have RNA processing, which we just talked about, slicing out the introns and putting the exons back together. All of this happens in the nucleus. Then that mature RNA leaves the nucleus, goes into the cytoplasm where it joins with the ribosome. And then the messenger RNA is translated by the tRNAs to produce a polypeptide. And then finally, in the cell, you will have the post-translational modifications. A lot of times in the Golgi is where this occurs. And then that is your big picture of protein synthesis. Question 13, describe the events taking place during interphase and their functional significance. So you can get a good idea as the figure 3.33 in section 3.8. So remember, interphase is made up of G1, S, and G2. With G1, this is where you're going to be growing new organelles, cytoskeleton, cytoplasm, getting ready So you're preparing. During S phase is DNA synthesis and replication. And then in G2, further growth and preparation for division. So G1, S, and G2 are our 3 phases of interphase. And then with questions 14 and 15, DNA synthesis described in figure 3.34, helicase unwinds, DNA polymerase, primase lays down the primer, DNA polymerase comes in, lays down the nucleotides, and goes along both strands. We have a leading and lagging strand, and at the end, your 2 new strands are semi-conservative. So they're semi-conservative replication, where each new strand is made up of 1 old, 1 new, so you have built-in proofreading with that. So that's question 15, figure 3.34. And then if you go on to the next, figure 3.35 will be our whole cell cycle. So you need to include interphase in the entire cell cycle to get full credit. So interphase, G1, S, G2. This is where the cell is growing and replicating the DNA, preparing for division of the nucleus, and then that's mitosis. And so remember, mitosis is just a very small part of the cell cycle. Majority of cell cycles in interphase, and mitosis is this little bit with cytokinesis. And then they're described in detail here. Prophase, chromatin condense, nucleolus disappears, nuclear envelope disappears, mitotic spindle starts to form. Then in metaphase, those chromosomes are lined up on the mitotic plate, as we looked at in lab. And then, I don't need to go. And then once they're lined up on that mitotic plate, we move into anaphase. This is where these sister chromatids are separated from each other. The mitotic spindle pushes the ends of the cell further apart, elongating the cell. And then telophase, exact opposite of prophase. Nuclear envelope reforms, chromosomes uncondense into our chromatin. And then we have the cleavage fur starting to form. And then we end with cytokinesis, where the 2 cells are pinched in half, giving us our 2 new genetically identical daughter cells. So that's your answer for 14 in figure 3.35. And then the last, compare meiosis and mitosis and their outcome. And so with mitosis, the purpose of mitosis is for growth and repair. And so you get genetically identical. So the result of mitosis is 2 genetically identical cells to the parent, and that is for growth and repair, whereas meiosis results in 4 daughter cells. Remember, with mitosis, they're also diploid because they're genetically identical. With the daughter cells of meiosis, we're going to have 4 daughter cells with only half the chromosomes, so they're haploid, and you also had the genetic recombination during prophase 1 and separation of the homologs in metaphase 1. And so they are genetically unique also. So those are the differences between mitosis and meiosis. So hopefully this has helped you with Chapter 3 to review for Exam 1. And I will try and get you a review for Chapter 5 out. If you have questions, be sure to let me know. Have a great day.